so much. It's a great pleasure for me to share my data, to share my research to such a novel audiences. Thank you so much to the OMICS group organizer. I'm sorry I have a bad throat though, but I will try to make up today. But basically I am trying to present is microRNA. So microRNA is important because they're small. They regulate gene expression by interfering their transcription. And this is 23, 27, 24. There are three members in one cluster. They finally regulate bone formation. So a couple of minutes ago, I listened that therapeutically osteoarthritis is very demanding and challenging. I absolutely agree. Because when we look at the arthritis at the end of the whole disease processes, it is very difficult to understand what is the molecular mechanism behind that whole arthritis processes. So I will shed some light, if my research permits, on that way. So basically, the non-coding RNA, they do not code for proteins. So this was like last 20 years, early 90s, non-coding RNA was not at all. People don't even know the central dogma was DNA, RNA, and the protein. Now already the NIH, because of their efforts, they almost sequence 70 to 90% human transcriptome. And they found only 2% they code for protein. And that account 21,000 protein coding genes. What happens to 9,000 plus 32,000 plus another 11,000? This non-coding RNA, huge population of mammalian genome is comprises of is mainly the structural RNA, transfer RNA, microRNA, small interfering RNA or siRNA, nuclear RNA, nucleolar RNA, the viral interacting RNA, promoter associated RNA, and then finally long non-coding RNA. And one of the RNA is in the pipeline, that's the gist RNA. It's a single RNA that can recruit and that can modify the whole chromatin. That's the new concept to develop potential therapy, RNA therapy for all human diseases. And we know these small RNA are so important in disease processes, in development. We cannot forget these four scientists. Victor Ambrose and Gary Rubcom in 1993, they were in Massachusetts, MIT, Massachusetts Institute of Technology. They first discovered animal microRNA. It's just very small piece of RNA of 22 nucleotides, but any point of time they can interfere the gene expression. Now this Craig Mello, where I work, he was in my Department of Cell Biology, and Andrew Fire from Stanford here. They discovered in 1998 how genetic interference you can generate outside the cell and put inside the cells, and you can regulate the gene expression. So these four guys are so important in RNA biology, small RNA biology research. And this, in 1998, when they discovered this, 2006, they got Nobel Prize in Physiology for RNA interference. Now this is very simple. MicroRNA is very, very simple. So this is host gene. And the microRNA might have their own transcription or might be embedded with the gene, any coding gene. See, this is the intronic red portion, this is the intronic sequences. After splicing, this microRNA forms such a primary microRNA, and they are processed by another RNase enzyme, is called dicer. At the same time, there are enzymes in the nucleus called DROSA, but the DROSA dependent processing, and this is DOSA independent processing. I don't want to make things very complicated, but what happens, finally from this transcription unit, they come a single stem loop RNA structure that is processed in the cytosol or in the cytoplasm by another RNase processing enzyme, which is called dicer, and it processes a perfect 22 nucleotide RNA. Now see their diverse function. They can target this gene, they can target this gene, they can target this, all this A, B, C, D and everything, multiple level. So if we can find the disease pathology here, right here, that is instituted by multiple target genes, we can take this single microRNA to control all of them. That's probably the best therapeutics probably coming in the near future. And now how they function. So there is a complex, it's called a RISC complex, RNA-induced silencing complex. And then RISC complex is a complex. Do you know that's the globular protein? It's called silencing proteins. This is the microRNA and this is the messenger RNA. If they have complementarity like 100%, they can kill the mRNA right there. If they do not have complementary 100%, they can block the translation. So both way we can control the expression of this gene which is harmful or which is making the disease or which is modifying the disease at any circumstances. So it is very proven that how microRNA regulates our skeletal system. When we knock out dry cell, when we knock out argonaute protein, these are all individual components to form bone. And this is a very, very tissue-specific deletion we did with osteocalcin gene that's expressed only in bone. And you will see in 8 months, the bone is 10 times higher. So what it means? That means that microRNA processing our bone formation. And another thing is there are some inhibitor that is functioning in presence of microRNA, and then microRNA just regulate them. That's why we have high bone formation. So if we can dissect these whole events, we can find a gold particle that will help to develop our new therapeutics for RNA. So, and this is a very, very hard fact, like from the morning I'm listening that 40 million people, so 40 million Americans are affected by arthritis in 2008. And we are estimating then 60 more millions in 20 years. So it is a huge burden because of our more life expectancy at the same time aging. So we need to do something. And this disease is not a simple disease, like not E. coli or not staphylococcus. It is a multifaceted, it is very complex, and a lot of tissue involved there, bone involved there, joint involved there, cartilage involved there. So these things is really needs very, very much attention. Now, if you really this is my view of seeing osteoarthritis. So all the risk factors are here: aging, obesity, mechanical stress, inflammation, genetic changes. In addition to that, your lifestyle. And like if you go in more molecular, if you just put this microscope there, you will see everything is regulated by genes, and that gene is regulated by transcription factors. On the other hand, I give more emphasis on this one because epigenetics is a big role now playing in all disease processes. Faulty processing or faulty mechanism is either upregulated or downregulated of a particular class of microRNA. If we find one of them, maybe we can switch or divert from catabolism to anabolism, and we can make a better lifestyle, we can make a better choice for mankind. So now I'm giving a simple example. This is called MicroRNA 140. And now in one way it's making the cartilage or chondrocyte homeostasis. On the other way it's making a balance in endochondral bone formation. Now if this is the target list, 1, 2, 3, 4, 5. Now somehow by modulating this one, can we modulate these processes to make prefer anabolism rather than catabolism? So that's my goal. Taking to that goal, and this is a list of whole microRNA, like this SAT31. This is a chromatin modifying enzymes. Now they are in the pipeline for aging, apoptosis. And then all these target genes are so important, they are regulating the microRNA in the development of OA. My favorite was 27A and 27B. So now I discovered after coming back from Massachusetts to University of Alabama, I was working in my own laboratory, and I try to just like precipitate. This is called a precipitation. So in your tube, you can precipitate the silensosome. Now silensosome basically contains the protein which is responsible for the silencing activity, microRNA and the messenger RNA. You can have all of them. Then you can go from here to a 3rd generation or 4th generation RNA sequencing or DNA sequencing. You have their all molecule identified. You can analyze them. You can go for further target analysis, and you can do whatever you want after that. So this is the samples Dr. Jun Lee, he gave me, because his department is osteomyology and our department is, I work in the School of Dentistry. So he found the synovial fluid from RA and OA patient. There is a new concept growing on is the fluid has a vesicle, it's called exosome. So I simply isolate exosome to see whether microRNA is there or not, or silencing machinery is there or not. See, basically, we use non-specific immunoglobulin and the globulin against the Argonaut protein, and we saw the silencing complex is present in the synovial fluid. And then I make RNA from the synovial fluid, and we found microRNA 23, 24, 27, the whole cluster with a very high C2 value they are expressing there in the synovial fluid. So what they are doing? Now I'm sorry for these slides though, but this is a osteoblast or bone differentiation. So a program of gene transitioning from one stage of cells to another stage of cells. Suppose this preosteoblast, before that this is the stem cell population or mesenchymal stem cells. They go all the way from osteoblast to osteocytes, and see a series of gene expression happens there. And a particular stage, this profile of gene expression predominates over this profile of gene expression, and see each and every stage. This is all the reported data I compiled, and I saw like each and every stage of this bone differentiation, microRNA and transcription factors, they are so much networking, and that's the result. The whole developmental process or whole differentiation process happens from stromal cells to osteocytes. Now this paper I published in 2010 in PNAS, and then later we wrote a Nature Review article over there. So this is a very, very complex circuitry. This cluster regulates two important candidate molecules that differentiate osteoblast differentiation, that play a very major role. But this microRNA, on a negative feedback, they regulate one of the very important tissue-specific factor, RNX2, and another homeodomain transcription factor, A10. So this is very, very tightly regulated. When I overexpress this microRNA, I saw they're completely blocking of bone formation or delaying of bone formation. So then I thought this is very right choice to develop mouse model on that. So I developed mouse model in two different approaches. Number one, I just globally knock out the whole locus of the cluster and see what is the phenotype. And the second one, okay, so I'm going, okay. So then I saw these phenotypes has much more bone, even though they are heterozygous mouse model. Unfortunately, I didn't get any homozygous because probably they are dying, they're lethal, embryonic. So see the differentiation in the bone formation from this to this, they have more bone and high bone mass. Now the second approach is even more noble. Like I overexpress anti-microRNA, that's the anti-sense microRNA, to block the function of the microRNA. And there is a tetracycline inducible element, I put it here. So if I feed the mother with tetracycline drug, the progeny will be suffering or deleting this microRNA right there. So I can analyze different stage of development, embryonic development, at the same time I can do the postnatal bone formation. So this approach, I am overexpressing anti-microRNA to block the function of the microRNA, and the other approach, I just completely knock it out from the genomic locus. But the result of both the mouse model is there is more bone formation. See all the bone density, bone formation, all the osteoblast number, and RNX2 and ostrix, these two markers, they went up. So this microRNA is inhibiting bone formation. So if I use an anti-microRNA, so we can use to develop a therapy that's 22 nucleotides, so I can make more bone when we need. And then, so in multiple approach, I need to come to a consensus that how important this microRNA is. So this is called high throughput RNA sequencing. So I make stable cell lines with this microRNA cluster, and then I analyze. We got like total genes of 2,705. They're more than twofold increase. And I am very much interested on the chromatin binding proteins, because that's, I think, the novel approach that is coming very in near future. So, and then I went back to mouse again, and I saw all these chromatin binding factors. They're important. They're so much upregulated. That means these if you knock down this microRNA, they will favor bone formation. And then I compile all this data with my RNA sequencing data, and then knockdown data in long bone, and then knockdown data in the calvaria bone formation, the membranous bone formation. I saw these are the molecules very, very important regulating the bone formation, and they are all the chromatin modifying enzymes. So I am I'm analyzing them, and I am going for chromatin modifying things, and that will be, I think, my ultimate choice. So this is a general conclusion that this cluster knockdown mouse have high bone phenotypes, down-regulates osteoblast growth and inhibit differentiation. This is this tiny biological process, RNA, repress gene expression may represent a power approach to treating the skeletal disorder, and including osteoporosis and osteoarthritis. And this is my sincere thanks to all my lab members, my departments, my collaborators. Thank you so much for your attention. Any questions?
Excellent. Thank you very much, Dr. Hassan, for a very comprehensive and interesting talk. Any questions?
You study mainly the microRNA from the bone, no? Why didn't you study it from the cartilage in the different stages of the degradative process?
Yeah, so I joined like 3 years ago in my new department. I do not have enough manpower, but that's I have in my mind. I am working in 3 aspects: the craniofacial development, because my school, and I am collaborating with Dr. Lou Bridges for the synovial fluids and develop some kind of therapeutic.
In what model?
I have a collaboration of University of Pennsylvania. They have make a mouse called PGR4. That's a proteoglycan on the cartilage. So they knock that out, and I am currently analyzing their RNA-seq data, whether that can fit with my chromatin modifying enzymes. Then we can go for, because one of the molecule is called PHF10. That's a chromatin modifying enzymes. Whether it is animal, whether it is mouse, or whether it is human, PHF10 is the same. So if I found PHF10 is potential, and I request Jackson Laboratory to have that mouse in my laboratory within 2-3 months. So that will be very interesting to study the cartilage development as well as the bone development in that way.
Yes, because we're studying the destruction of a cartilage, so maybe we can do something together.
Yeah, absolutely, absolutely. I'm very open for
it. And we're just finishing all the molecules, well, we'll see it afterwards, that are participating since the beginning to the end in a very cheap model. It's not very expensive. So we could see together how we can determine the microRNAs of the molecules that are the one that starts the process.
MicroRNA data, I am a microbiology researcher, but microRNA data somehow very confusing if you do not validate like in a gold standard way. And I am very much interested in the chromatin, because that opens the chromatin for transcription. Mm-hmm. I do not want to go beyond that, because I want to see what happens right there at the level of chromatin. And some of the chromatin like HDAC inhibitors, then SAT2, SAT1, they're on the pipeline, they're giving very good results. I read in Nature Review Drug Discovery. Mm-hmm. So some of the chromatin modifiers might be very, in my knowledge, might be very, very important to deal the disease in future. I think so.
Okay, well, afterwards we can continue talking about that, because we have also some experience in bone. Oh. And now there's a contradiction, because people thought that the bone was the first one. But we just published an article showing that it could begin from this cartilage ossification process, from the cartilage to the bone. But, well, that's something that has to be well discussed.
Absolutely.
So thank you very much.
Yeah, thank you.
Thank you very much. Any other questions?
Hassan, in your microRNA knockout mice with that whole cluster gone, did you check for OA development in the joint?
No, the global knockout mouse was, I didn't get any homozygous. The second, the Jeep mouse I call, that is the anti-MIR mouse. When I treat with doxycycline, there were some problem, they were dying. This mouse I got like 3-4 months ago, so I am definitely looking at their joint to see how their normal joint versus the knockout joints are. I'm going to see that.
How does your work relate to bone morphogenic protein? Are these linked in any way?
So bone morphogenic protein, I consider that one as a signaling, Wnt signaling, bone morphogenic signaling, or TGF-beta signaling, and then you have Notch signaling, all this. So first of all, when we test any microRNA, we just treat the cells with bone morphogenic protein to see what's the expression of that microRNA, whether they are responsive to BMP or not. All three microRNA, I already published that they're responsive to BMP. So initial induction, there is a fine mechanism I describe in my PNS paper, that microRNA, they express maybe across the different, like muscle, bone, fat, all. And some of the common microRNA you might find, but the tissue-specific microRNA is somehow controlled by a tissue-specific factor and a particular signaling. So BMP Runx2 regulate this cluster of microRNA to make a bone kind of programming. And that I saw in my PNAS paper. So they are responsive to BMP, and they are regulated by Runx2, the tissue-specific transcription factor. So the crosstalk between a tissue-specific transcription factor and the signaling works right there to make that microRNA tissue-specific or not tissue-specific.
Okay, any more questions?
Just one question. The questions between the subchondral bone or articular cartilage in the initiation of osteoarthritis, if that debate still exists, now in your observations, where are the distribution of the microRNA among the knee joint, in the synovial fluid, subchondral bone, or articular cartilage, or somewhere else, or in general?
So I didn't address that in animal, but I took different kind of cells, like ATDC cells for chondrocytes, and then HIG82 cells for the synovial membrane, and I treat with the fluid, either osteoarthritis fluid or arthritic fluid. I saw the modulation of the same microRNA over there too. Yeah, same distribution.
But ATDC5 is a cancer cell line. Why don't you use chondrocytes?
No, ATDC5 is a chondrocyte. ATDC5.
It's not a chondrocytic cell line. It's a mouse.
It's not articular chondrocytes, but it is a chondrocyte. ATDC5.
There are no chondrocytic cell lines except the ones which were developed by Mary Goldring, and those are all transformed chondrocytes.
Yeah, that's true. So it's better to freshly isolate the primary from the articular gut and see the effect over there.
Any more questions?
Okay, excellent. Thank you very much.